I'm Stefan Alexander. I'm the E.E. Just professor in the physics department here. Welcome to the second keynote address. I want to invite our what's the word? The dean I love so much, the dean of the College of Students, Charlotte Johnson, is going to introduce Professor Langford.
Thank you, Dr. Alexander. When I first arrived at Dartmouth a year ago, I heard repeatedly from students about the importance of the E.E. Just program and the need for its continued growth and development. Dr. Alexander's appointment as the E.E. Just professor and the symposium are both wonderful markers for our community. But enough about Stefan. I knew that would get your attention. It is with great pleasure that I introduce Dr. George Langford and welcome him and his lovely wife, Sylvia Langford, back to Dartmouth. Dr. Langford was our first E.E. Just professor and director of the program. Since 2008, he has been the Dean of the College of Arts and Sciences and professor of cell biology and neuroscience at Syracuse University. A nationally known neuroscientist, Dr. Langford received his PhD in cell biology at the Illinois Institute of Technology in 1971 and was a National Institute of Health Fellow at the University of Pennsylvania from 1971 to 1973. Dr. Langford's research interests include cell and molecular biology and neuroscience with medical applications to treat Alzheimer's and age-related conditions. In 1998, Dr. Langford was nominated by President Clinton to the National Science Board, NSB, the governing board of the National Science Foundation, to advise the President and Congress on national science policy. He served on the NSB from 1998 to 2004, as chair of the NSB Education and Human Resources Committee from 2002 to 2004, and as vice chair for the NSB National Workforce Task Force Subcommittee from 1999 to 2004. The E.E. Just program seeks to engender scientific excellence for all Dartmouth undergraduate and graduate students, with an emphasis on underrepresented groups in the sciences. Like Ernest Everett Just, Dr. Langford left an indelible mark here at Dartmouth, and we are so pleased to have him here to share in this exciting time in the program's history. He is a trailblazer and our own piece of African-American history. I bring you Dr. George Langford.
Thank you so much, Charlotte, for that very kind introduction. It is such a pleasure to be here. It's wonderful to see old friends and colleagues, to be back on a campus where I really had a wonderful, wonderful period of my life. It's also important to be here for this particular occasion, to be part of a symposium that is designed to celebrate the legacy of Ernest Everett Just, but also the inauguration of Stefan Alexander as the next E.E. Just Professor of Natural Sciences. So I'm really, really proud to be here and to participate in this momentous event. It's also great to see all of the students who are in the audience, and to hopefully I'll have some words of wisdom to pass on to you as well. I can remember when I first arrived. I came here in 1991 as the Just professor, and it was a wonderful occasion. At the time, Jim Wright was the dean of the faculty. Jim has been a wonderful colleague and friend over the years, and he asked me to give the public lecture after my appointment of the E.E. Just professor. And I can recall in the audience there was Gary Love, a treasured alum of the college, and Trustee Lyle Carter was also in the audience. And they really welcomed me and made me feel a part of Dartmouth College. So it's one of those moments that I treasure in my own career. So I thought I'd do a couple of things today. I wanted to first talk a little bit about the importance of Just life to my own career, and then to talk a little bit about the eJust program and the work that still needs to be done. And finally, I will review my own research while I was the eJust professor here at Dartmouth College. So, as Stefan mentioned yesterday in his introductory remarks, eJust was a hero in the black community and represented one of the best and the brightest. And while the public is most familiar with African American intellectuals who contributed to music, art, and literature, names like Zora Neale Hurston, Langston Hughes, Duke Ellington, these names are very familiar to everyone, but Just was part of a small but very significant group of scientists who participated in the Harlem Renaissance during the 20s and the 30s. So he was a major contributor to the movement that was part of ensuring that the world understood that African Americans could contribute to not only the arts, but to the sciences. And of course, today, Just's story is very well known because of the work of Kenneth Manning, who wrote his biography, The Black Apollo of Science. And we now know of both the accomplishments. I think the world is familiar now with both the accomplishments, but also some of the very, very difficult experiences that eJust had. But I always marvel at this, at his life story, because as a young boy growing up in Charleston, South Carolina, being able to leave his home to go to Kimball Union Academy, and then to do extremely well at the boarding school and to earn admission to Dartmouth College. So he came here, graduated in 1907, and then went on to the University of Chicago to do his PhD work. His career was significantly influenced by his affiliation with the Marine Biological Laboratory in Woods Hole. And I want to talk a little bit more about the MBL later on, because that is also a place where I did a great deal of my own research. And so I do share in common with Just the experience of working at this incredible place, the Marine Biological Laboratory in Woods Hole. So just briefly, Stefan touched on this yesterday, but as you know, Just made seminal contributions to science. He perfected the technique of in vitro fertilization using the sea urchin eggs. This was the first time that one could actually fertilize eggs in the petri dish and watch early stages of development. So it was a major development at the time. But Just also went on to develop other techniques as well as contributions to science, including his real emphasis on the fact that the cytoplasm played an important role in the development and the phenotype of the developing embryo. And again, as Stefan mentioned yesterday, there is this new emerging field called epigenetics, which was one of the areas that Just made contributions to in his career. So anyway, I feel very, very privileged to have been the eJust professor, and I feel that I stand on the shoulders of him because it was because of his persistence in science that my career has been very different. Whereas he had to actually prove that blacks could, in fact, make original contributions to science and the scientific literature, I've had a different experience and have been able to pursue my career because of the advances that he made to science. So again, I'm delighted to be here today. I wanted to pass along a gift to Stefan as a token of my admiration for him and the work that he has ahead of him as the E.E. Just Professor. So to Stefan, I want to give you a copy of the book that Just wrote. This is a reprinted version of it, but it is the biology of the cell surface, and I'm very, very pleased to give that to you. So I'd like to just talk a little bit then about the E.E. Just program, because the E.E. Just program was something I was very proud of to be able to start and to pass that on to Stefan to carry forward. But it was designed really to increase the number of students, African American students in particular, who majored in the sciences. Because one of the things that we have always known, and we still know this to be the case, is that students enter the college, Dartmouth College, with an interest in science. When we polled students back in the early 90s, we noted that for African American students, the same number, same percentage of students had an interest in science as white students. And that was about 45 to 50% when they enter the institution. But upon graduation, about 30% of white students majored in science, whereas it was about 15% for African American students. And so we really wanted to try to bridge that gap. And we knew it was not because the students didn't have the ability to do it, but there are other structural factors that prevented students from pursuing their own interest. So the program is really designed to set up to address some of those issues. And we set up the Science Forum and invited students to come to hear professors talk about their research. And we wanted to actually provide opportunities for students to get to know faculty members and for them to hopefully do internships in the faculty laboratories. And I recall one of the funny moments was when I first started the Science Forum, I decided that we would do it as a Saturday morning breakfast. And if you know anything about college students, Saturday morning was not the time to schedule a science discussion. And so we learned very quickly that we could do a luncheon, and hopefully by that time, students would be up and ready to come out to participate in this program. And I want to give a shout out to my wife, Sylvia Langford, who was part of this whole process. She was always helpful in designing the programs and participating and coming out and really encouraging the students. So to Sylvia, I want to say thank you very much. So, the problem continues to exist. I was recently invited to participate in a conference that was hosted by the Howard Hughes Medical Institute and the National Institute of General Medical Sciences at the Howard Hughes headquarters in Chevy Chase. And it was on this topic of improving underrepresented minority persistence in STEM. And so we've spent 40 years really working on making sure that students can pursue their interest in science. And we still have a lot of work to do. But I was pleased to be part of this panel that was discussing this problem. And the funding agencies, the HHMI and the NIH, are interested in finding new ways to really address the problem. So I wanted to share with you some of the ideas that came out of this conference. we began by just thinking about what are the qualities that make for a scientist. So what makes a great scientist? And we heard a wonderful lecture from Jim Gates yesterday, who's a theoretical physicist. And there are qualities that are required if you're going to be a theorist, and other qualities that are required if you're going to be an experimentalist. But all of them include these kinds of factors: imagination, curiosity, risk-taking. And the question becomes, how do we tap into these qualities that students bring with them to the college? We know that students are naturally curious, and they have determination, they have interest. So we can impart scientific knowledge, but we also have to help them to tap into their own resources. And one of the analogies that we have used in the past is this idea of a pipeline. We know that students start out in elementary school with a great deal of interest in science. By the time they get to junior high school, we see fewer kids, and then high school, there are fewer yet who have an interest in science. And we use this image of a leaky pipe. And what we thought about at the conference was that perhaps we should change the analogy. We should not so much think about the pipe as leaking, but think about the students. And if we think about the students as water that needs to be lifted to the top of a mountain, that provides the energy that allows them to persist when the times get difficult. So maybe we can use a different analogy. It's the water on the mountain. So programs should be designed to lift students, to motivate, to inspire, to find their own creativity and joy. And I think programs of the future will begin to develop these kinds of qualities to make sure that students persist. So creating lift. After 40 years of interventions, we can now identify practices that institutions use to do this. And I think the EJUST program is one that can definitely do that. But other programs that we can think about around the country would be the Meyerhoff program at the University of Maryland, Baltimore County. Programs that really have shown to be extremely successful because they know how to motivate kids to make sure that they can persist in the discipline. So what we did as a group was to create this kind of a matrix. We said, this is perhaps the way we can begin to think about this whole issue. If we think about programs that addressed what we call the low-hanging fruit, those kids that are highly talented and prepared, and that would definitely represent the students who are coming to Dartmouth College. There are also higher hanging fruit, underprepared, but students with great potential. And then look at both individual skills as well as the contextual level, such as the activities of faculty in the institution. And then we can fill in these quadrants of different programs and decide, identify those programs and the features that make it work, and then how do we scale up those programs. And so again, I think for the EJUST program, it would be one in which you have highly talented students, but there are contextual issues that need to be addressed to ensure that those kids are successful. The Meyerhoff program would fall into that, but there are many programs that work with kids who are underprepared but have great potential. And the question is figuring out how to make sure that we can scale up those programs that are successful. So just a set of recommendations that came out of this conference was to really think very seriously about the curriculum. This is one of the factors that really makes a big difference in the ability of kids to succeed, especially the gateway courses. We have to change how we deliver content in those courses, changing the content and the epistemology. We are doing a lot of work at our institution with new kinds of pedagogy in which we use cases where kids can actually watch videos before they come to class so that there is time in class, in large classes like this one, where kids can have discussions among themselves and with the faculty members. And this has been a very, very successful program. And it's actually the physics department that is leading this effort. They've done much more with changing pedagogy than I find in some of the other science departments at Syracuse University. So changing the pedagogy, really getting kids involved at an early stage in asking questions and thinking like a scientist. So model for students how scientists think. Then the research experience, which everyone knows the hands-on experience is one of the greatest ways to get kids excited about science. And then really helping faculty to change the way they present their materials to students. And finally, partnering with the standardized test makers, because they drive a great deal of the way content is delivered to students. And then this is a favorite of mine. I think we really have to understand that it's a national imperative to increase the number of students in the STEM disciplines. And so what we have to do is to get students to understand that they are part of a movement. There should be a presidential level announcement that the country is dedicated to increasing the number of students in the STEM disciplines. And if you can motivate them so that they understand that they are part of a greater good, to really support an activity for moving the country forward, much like the race to the moon, then I think we can also really captivate the interest of students and help them to persist in disciplines that are very difficult. So, as a go-forward guiding principles, institutional transformation, we have got to figure out ways to really help institutions change the way they deliver content to students and provide the kinds of support that they need, and then really helping to establish this idea of a culture of success. So these are all of the things that I think we still need to do, but I think it's one of the things that the EHS program will provide for students here at Dartmouth College. So with that, I'll move on to a review of my own research. And much of this work was done while I was the EHS professor here at Dartmouth College. But I wanted to describe for you the work that we do on transport in nerve cells. We use the Squid giant axon as a preparation. And as I mentioned before, this work has to be done at the Marine Biological Laboratory because we need freshly caught squid. So it was an opportunity for me to go to the MBL each summer, and I would take students with me. It was a great experience for training students there, as well as for getting research done. So, but anyway, this is my general area of interest is in the brain and memory. What are the mechanisms, the cellular basis of learning and memory? But our real work deals with the individual neuronal cell and the process by which materials that are synthesized in the cell body are transported along this very long, thin structure called the axon. The nerve cell is quite unique in its structure. It has what is called the cell body, where the nucleus is located, but then this very long, thin axon that leads to the nerve terminal out here. And most of the protein synthesis occurs in the cell body, here around the nucleus. And you can see that there's a very long distance that materials that are synthesized here have to travel in order to get down to the nerve terminal. And this process cannot occur by simple diffusion. So we know that if you had to depend upon diffusion for materials to get from here to here, it would be a very, very long time. So instead, the cell has a very, very highly developed transport system. These are filaments, part of the cytoskeleton, microtubules and actin filaments that are aligned along the axon that function to transport materials down to the axon terminal. And you can see the differences in length. Some axons are very short. Others can be up to a meter in length. So this is a very, very long distance that they have to travel. And so my interest has been in the process that we call axonal transport, how you transport these proteins and actually membrane-bound vesicles, which I study, from the cell body down to the terminal. And of course, there has to be the reverse transport as well, because you have to maintain the normal volume of the terminals. And so you have a two-way process. We call it anterograde from the cell body to the terminals and retrograde from the terminal back to the cell body. So that's the process that, that we have studied. And then down at the terminal, the axon forms this bouton that is filled with a small number of vesicles. These are called synaptic vesicles. And this is where the electrical potential that travels down the axon is converted into a chemical signal, which happens because these vesicles fuse with the membrane and release their content, the neurotransmitter, into this gap here that excites the next cell. So it's an electrical activity that gets converted into a chemical signal, and that is what activates the adjacent cells. So the synapse then is this very important complex at the end of the nerve cell that provides cell-cell communication. So let's look at how this whole process works. Using the squid nervous system, you know, the squid it's a very interesting animal. It has a very complex behavior, but it's also very tasty to eat. So it's got lots and lots of predators. So we like to eat it, but it also has to always escape from predators in the wild. And so it has a lot of defense mechanisms. It is able to change colors. It has a very interesting pigmentation, so it can mimic the background and disappear that way. But it also, these are the tentacles here. It has a siphon here and an ink sac that it can also throw up a sort of a defense by spraying ink, and so it can hide from the predator. But the other thing that it does is the siphon is also connected to a sac that is filled with seawater, and it can contract its mantle muscles and squirt out the water to propel itself backwards. It's sort of like a jet movement going backwards. And so it can escape the prey that way. And so the squid has developed this phenomenal nervous system that allows it to conduct action potentials very rapidly, to contract the mantle muscles, to eject the water, to allow it to escape very quickly. So we take advantage of this nerve fiber that is called the giant axon. So remember, it's not the giant squid. We're working with the small squid, but it has this large diameter nerve fiber called the giant axon. So why do we use this? What are the advantages of the squid? Is this is one of the few preparations in which you can actually view the activities that are taking place inside of the cell after you've removed the cell membrane. So we can actually have direct observation using a technique that we call video microscopy. And this was a technique that was developed both by Robert Allen, who was a faculty member here at Dartmouth College, and Shinya Inoue, and was one of the advances in microscopy that really propelled our field forward during the 80s and 90s. So we can observe this activity directly. We can do this in vitro. That is, we can actually take the cytoplasm out, put it into a buffer, an artificial buffer, and that maintains the activity in vitro, which is pretty unique. We can't do that with a mouse axon or with any other kind of axon to date. And so we can do real-time analysis. We can reconstitute a lot of this activity with purified components. We can add inhibitors, such as antibodies or recombinant proteins, to block activity, and we can also purify these proteins. So it's a pretty robust preparation to work with and really exciting. So this is just a quick overview of the nervous system. There is the squid has a small brain. It's called the optic ganglia because it's primarily responsible for controlling the eyes. But there's also this stellate ganglion that gives rise to this large fiber called the giant axon. And this is the nerve fiber that we take out. What is interesting about this fiber is that it represents the fusion of many individual axons. So in our bodies, the cell body gives rise to an axon, and the axons, each individual axons remain separate. Here we have 1,000 cells that put out axons, and then the axons fuse into one. And so we have then this large diameter axon. And the reason that this is important is because the cross-sectional area of the axon determines the speed of conduction. So the larger the diameter, the greater the speed of conduction, and that is how they're able to conduct these action potentials so rapidly for contraction of the mantle. So again, the cell bodies are here. They fuse to give you this large diameter fiber. Here it is, the giant axon. And you can see this is the interior. It is surrounded by connective tissue. And that's the preparation that we dissect out of the animal. So we have to do first a gross dissection and then a fine dissection. And then finally, we actually remove the membrane. And that's the real trick of this preparation. And we end up with a whole cell extract in an artificial buffer. And that's what we see. So in the summers, I would take students with me to Woods Hole and train them on how to do this preparation. You open up the animal. You can identify the giant axon. We take it out, put it onto a glass slide, and extrude the exoplasm. And this just gives you a view of what it looks like. Here is the stellate ganglion. There's the large fiber. And you can see what we do is lift it from the surface of the mantle. We tie it off at the two ends and then cut. So there it is, the fiber with threads tying off the two ends. You can see the diameter. It's about 1 millimeter in diameter. And then the fine dissection to remove the connective tissue. You have to be extremely careful not to damage the cell membrane, because if you do, then the activity dies. And here we actually use a Teflon tubing to remove the membrane. We simply push it out the way you would toothpaste from a toothpaste tube. And we end up with the small little piece of exoplasm that we can then observe in the microscope. So let me describe just briefly how this microscopy works. We call it Allen Video Enhanced Contrast Differential Interference Contrast Microscopy, but it's a form of what has been described as super resolution. We can use the video to both enhance contrast and to detect particles and filaments that are below the resolution of the light microscope. So it's pretty incredible that this technique works. And we can actually detect microtubules as small as 25 nanometers, which is an order of magnitude smaller than the resolution of the light microscope. So a light microscope can usually resolve particles around 250 nanometers in diameter. We are detecting things that are 25 nanometers in diameter. And again, it's not resolving them, but detecting those structures. Actin filaments are too small to be detected, so we use fluorescence microscopy to pick those up. So this is the technology, and I won't go through the details of this, but we use the video camera to capture these images and a digital camera to use for the low light level imaging of the fluorescence images. And then we can do the analysis of those preps. So this is the way the contrast enhancements worked. If you were to look through the microscope with your naked eye, this is what you would see. There are bright spots. There are bits of debris or mottle that you can remove by subtracting a background. You see it's gone here. And then, so in real time, we do a background subtraction, and then we do a histogram stretch. We can actually identify the gray levels and bring out that contrast so that you now detect particles that are not visible here. So that's the beauty of that technology. And using that, then, we can do real-time video capture of the movement that's going on in these axons. OK, so this is what the preparation looks like when you extrude it. Here is the bulk axoplasm. But notice here, you see these small filaments and particles. And these microtubules here have dissociated from what we call the bulk axoplasm. And out here in this very thin region, you can begin to see individual filaments as well as individual particles. And in the background, which you can't see here, are these thinner filaments that we call the actin filaments. And we can only detect them by staining them with a fluorescent dye and viewing them using the fluorescence optics. So I'll show you a movie now of how this works. This is the axoplasm that we've taken away the membrane. These are the microtubules, vesicles, and actin filaments. And then some of them diffuse out of the bulk, and we can see that activity out here at the edge. So let me show you a movie of this. If I can get this to work. So this is speeded up about 10 times, but you can now see the level of activity that's going on. This is happening in every one of your nerve cells all the time. You can see very tiny things moving. Which direction do you see most of the small particles moving? That's right, to the right. And so we know that that's the anterograde direction. Most of the small particles are moving toward the right. The bigger ones were moving toward the left. So you have both directions. You have the anterograde and the retrograde movement. So then if we move out to the edge, you can see these individual microtubules. And notice that you can see particles that are moving on tracks that are invisible by this technique. And I want you to take a note of this particle here. So this is the time here in seconds. So it's repeating. It starts at 44, and then it repeats again. But notice what happens when this particle attaches. When it gets to the end of the microtubule, it then hops off onto the actin filament, which again we can't detect except by the fluorescence microscopy. So this was the novel observation. It was well known at the time that particles moved on microtubules. That was well known. But no one had ever observed the movement on actin filaments. So we were able to show that both filaments were involved in the transport of these vesicles. And what we proposed is that the microtubules served as the expressways. So long-distance movement occurs on microtubules, but when the particles get to the end of the microtubule, then they can hop off and go to their site of destination, out to the cell membrane or down to the terminal, a region where there are no microtubules. So just to summarize, again, you saw this particle as it was moving along the microtubule. I have it here in green, and then at the end it hops off, and the particle I indicated as red here as it moves on the actin filament. So the model is that long-range movement occurs in microtubules and the short-range movement on actin filaments. And now this is accepted as a general theory for transport of membrane materials in all kinds of cells, not just nerve cells. And we went on to identify the motors that were involved. The motor Kinesin, which is a microtubule motor, had been discovered in the 80s. And as a matter of fact, I think it was just this year that the Lasker Prize was given to the scientist who first identified kinesin. It was Ron Vale and Mike Sheets. This is a protein that binds to a vesicle by this globular tail end. It has a rod-shaped structure, and then the heads here. And we show that a motor that's responsible for movement on actin filaments is myosin 5. And it's a very different kind of motor. You can see it has globular heads like the kinesin, but it has these very long neck regions, and then a rod-like tail and a globular end here that again attaches to the vesicle. And so this is an animation of how we imagine this process is working. So a vesicle has two motors attached. We believe that those motors are interacting with each other, and there is some communication. So this is kinesin that's walking hand over hand along a microtubule. When it gets to the end, then it flips around and the myosin 5 motor begins to move. So you can see that the myosin 5 has these long neck region, which allows it to take much longer steps, but it is walking along this thin actin filament, carrying the particle to its final destination. So that's the dual filament model. And again, this is the work that was accomplished while I was here. And I want to thank the students. Many of these were students here at Dartmouth: Carl DeSelm, John Dela Cruz, J.P. Flores, Sung Yu, who were working with me on these projects, who spent summers at the MBL. And it was my postdoc, Phil Stafford, who did the graphics for that lovely movie. So with that, I will stop and take questions. Thank you very much.
Will promptly leave by 9:45, so we have a good 10 minutes, 12 minutes for questions. And I see lots of students, so we'll take questions from students first. Yes?
Do any other organisms be used to do the actin filament besides soy?
There have been other organisms that you can view the actin filaments. there are different kinds of motility assays that one can use. That is, you can isolate the myosin, the myosin motor that I described that moves the vesicle. You can isolate that, and you can actually attach that to a glass surface, and you can watch actin filaments glide over the surface. It's called a gliding filament assay. But no one has been able to actually reconstitute movement of vesicles like the ones that I showed you here. So that still has not been done. Yeah. Yes?
We're going to take some time for the microphone to go to...
Okay. All right. Good.
Hello. What application does your research, where can we go from here with what you found?
Great. Good question. So there are a couple of things. What we are interested in knowing is how electrical signals are converted into these chemical signals. That is, there are synaptic vesicles that occur down at the very end of the terminal, and it isn't clear how they move from one location to another. So we're hoping that if we had a better understanding of the process by which vesicles move on actin filaments, we'd have a better understanding of just how that signaling process works. So that's one part of it. But the other part is that when information is stored in the brain, there's a structural change in the connection, in the synapse, the connection between the two nerve cells. And so we think that these proteins that we've identified as being involved in vesicle transport are also involved in the remodeling of the synapse. So hopefully it will help us to understand the whole process of memory and what happens when memory is lost in diseases like Alzheimer's and other kinds of dementia. Good. Yes?
Thanks. Fascinating. Do you have any theories or have discovered what given that most motors or all motors run on energy, is it electrical energy that powers these motors, or is there some biological metabolism that happens at that level?
Thanks for that question. Yes. What we know is that these motors use ATP, so it's a chemical energy. So they're able to hydrolyze ATP, and the actual hydrolysis of the phosphate bond provides the energy for the movement. What is interesting about these motors is that they only move in one direction on the filament. So the filament itself is polarized. So depending upon the orientation of the filament, it determines which direction the particle will move. And then ATP binds to that globular head that attaches to the filament, and upon binding, it stimulates the hydrolysis of the ATP that releases the energy that allows it to go through this conformational change. And that's why it can do the stepping motion. So it's really chemical energy that's providing the source. Yes? Yes?
Thank you. George, I wanted to switch gears a little. As a biologist, you were talking about using the flipped classrooms at Syracuse, and at least what I've read is that a lot of the success with it happens to be more with physics and math, or at least it's been used more there. I wonder if you could comment about whether you've seen any sort of discipline difference in how useful it is, and also with your students. Is it more helpful for URMs or first-gen students, or is it across the board that you're finding it's a helpful approach?
Thank you. You're right that most of what we've seen has been with physics, but biology has also. I noticed that in the intro biology course that they use clickers, and they also have designed ways in which they can break the class up into small groups, have times during the lecture when students can have a discussion among themselves so that there is active participation, during the time of the lecture. And this works for all students. So it's very extremely helpful for underrepresented minority students, but I think it's a general technique that works across the board. What I've said to my faculty is that because students now come with their smart devices, they have their cell phones and iPads, that we have to figure out ways in which we can take advantage of that technology to help us to deliver the content. And I do believe that we haven't yet, optimized that process. And of course, you know that there are places like Stanford and MIT that are putting these MOOCs, massively open online courses, as a way to get more information about how students learn. It's not so much about online teaching, but it's really trying to understand, what are the difficult concepts and what are the best ways to present that content to students. And I think that will be the great advantage of this experiment that they're doing of making these free so that they can collect information using artificial intelligence software to get information about how students learn and what are the specific roadblocks to certain kinds of concepts. Yes, Jim.
Thank you, George, for that informative. Is this thing on? It is now. My question actually was about the very first part. So I'm switching with your colleague back to the first part of your presentation about the meeting that you described at with the Howard Hughes Institute and what have you.
Right.
As you know, I've been involved in some national level and presidential level investigations in this direction of increasing STEM enhanced performance of our teachers and learning by our students. Okay. And in the workshop that you described, what I'm really curious about is you talked about teaching pedagogy. How broadly was that discussed? Because as you know, you have, for this audience, you have demonstrated how elegant a professor can be in expressing ideas. And yet we have lots of evidence now that lectures really are not necessarily the best way to get students to learn. So I'm kind of curious about the discussion on pedagogy that you described.
Yeah. Well, it's a good question, Jim. And I think there is, at least among the participants in this conference, there was a sense that this discussion is going on all of their campuses. It's definitely going on our campus. And I think we are beginning to realize that we've got to change the pedagogy. And I think most administrations now are willing to invest some resources in doing this, because one of the recommendations that we said is that we've got to find ways to incentivize faculty to do it, because this is something that we really have to learn how to do. And so on our campus, for example, we are, what we hope to do is to bring in some experts to consult with us about what are the best ways to deliver content, and then set up workshops where faculty can come in and learn those new pedagogies. So I think it's beginning to catch on, and I think there is there's some competition, because we know that some institutions, some of the leading institutions, research universities, are already adopting it, and it's going to drive other institutions to do so as well. Yes?
The Vesicle Transport is a very elegant system, as you've outlined. Have you identified, or has anyone identified, the role of injury in this model, and whether it's a self-reparative process, and how does the Vesicle Transport system change in the face of injury?
Yeah, a great question. You know, there are actually some old studies that were done, right after or during World War II, because a lot of veterans were coming back with neurological injuries. And so they're very curious about what happens to this process when nerves are injured. And so they did some experiments. They could actually tie off nerves in the middle and see what happens at the point of ligation. And what they found is that materials accumulated at that point on one side, and they disappeared on the other side. So it's known that if you block the nerve, that it prevents the movement through that site. And if you actually cut the nerve, then the nerve dies back because of the loss of the ability for the system to continue to work. So it's another part of trying to understand processes like spinal cord injury, where once nerves are severed, they usually do not regenerate. It may be possible to have some positive effects on nerve regeneration if we had a better understanding of how these systems work and how we could hopefully, get them operating again or some way to protect them if a nerve cell is injured.